Hi again, Gen Bio. We are going to pick up this video where we left off with the last one. We are now going to start looking at land plants that are seed plants. So we had already done the non-vascular plants and the seedless vascular plants in the last video. So now we're moving up to seed plants. And as I had mentioned in the last video, this group is characterized by dominant sporophyte generation and a reduced, often microscopic, gametophyte generation. Okay. And it's comprised of two major groups, the gymnosperms and the angiosperms. So gymnosperms are going to be all of our conifers as well as a few others, so cone producing trees. And then angiosperms are flowering plants. They can be trees, they can be smaller plants that produce fruit and vegetables. So those are all going to be our seed plants. Few key points we've already established. They have vasculature at this point. So they have xylem and phloem. Xylem allows for water to be taken up from the roots and carried up to the rest of the plant. Phloem allows for the sugars that are produced in the photosynthesizing leaves to be brought to other parts of the plant where that energy is needed. As I mentioned, dominant sporophyte gametophyte generation is usually microscopic. And we have distinct male and female gametophytes, which we'll see. And then now, of course, key point here, we have seeds. Seeds are going to enclose, it's going to be like a protective coat that encloses the plant embryo, which is diploid at this point. It's after fusion of sperm and egg has produced the plant embryo. Is housed within the seed coat, and it's packaged up with a food supply to get that embryo started when conditions are right. So the seed can be preserved for a period of time, which obviously has a benefit for the survival of these plants. The seed can be dormant until the environmental conditions are prime for this embryo to start using up the food supply and producing the mature sporophyte plant. Like I said, we're going to look at gymnosperms and angiosperms. So first, gymnosperms. What this means is that they don't have a seed contained within a fruit, within an ovary, whereas angiosperms have flowers that hold fruits, and that within that fruit, enclosed within that fruit, is where you'll find the seed. Gymnosperms have naked seeds. They're usually on the scales of a cone, held within the scales of a cone, such as you'll see on our evergreen trees. So you look on the evergreen trees, you often see the pine cones or other cones, and within those are where you're going to have the developing parts that will form the seed ultimately. Okay. All right, so you will also find needle-shaped leaves. This helps to retain the water in a gymnosperm. So they're often evergreens. They don't have to lose and drop their leaves every fall as angiosperms do. Some examples we're going to look at today as well as in lab this week are the blue spruce, the Douglas fir, the eastern hemlock, and the white pine. So we're going to have you guys take a look at what the needles and the cones look like for each of these so you can distinguish between the four examples. Before I move on and specifically look at the blue spruce though, I want to direct your attention to the mature gymnosperm here. This is the mature sporophyte, and point out that there are shown here two different types of cones. So this is the ovulate cone. This will produce ultimately the egg. So you've got within the ovulate cone, held within the scales of the cone, you're going to have the cells developing into the female spore, the megaspore. So this is going to be housed within that ovulate cone, and that will then, of course, develop into the female gametophyte. So the megaspore itself now is haploid, has developed from meiotic divisions of cells within the cone. So you've got a megaspore haploid. It will undergo a series of mitotic divisions, so still haploid, to produce then the egg, which will be held inside this structure, which you might ultimately recognize as the seed. So it's often on some plants, you'll see like the little twirly winged seeds that can then allow for dispersal of that seed to a location further from the parent plant. But before it's been fertilized, the female gamete is going to be developing within there. So the egg is going to be developing within that megaspore. And then on the male side, the counterpart here, you also have a pollen cone. This is where you're going to get ultimately development of the sperm, which will fertilize the egg. So within that pollen cone, we've got the microsporangium. So that holds the microspores. So again, remember our prefix is mega, always refers to the female parts. Micro always refers to the male parts, okay? So within the microsporangium, there are cells that undergo meiosis to produce the male gametophyte. So the pollen, each of the pollen grains then that are released have our male gametophyte within there. And then there are cellular divisions, mitotic divisions, which will maintain the haploid number and produce sperm. And then of course, at fertilization, sperm meets egg, and then you restore the diploid number. So now we've got our embryo, which is enclosed in a seed and can be packaged up and maintained until conditions are right for that seed to germinate. And then when it does, it will develop into the sporophyte generation, which we would recognize as the large tree. Okay. All right, so let's look at blue spruce now in particular. So blue spruce are characterized by a much more blue coloration to their needles compared to other evergreens. So as you'll see here against the green, the evergreen background, you can see that the blue spruce takes on a much more blue tinged look to it than many other evergreens that you'll see. Additionally, the needles are short and very sharp. So they are, if you touch them, rather pointy to the touch. They're about 3 centimeters long, and they are attached to the stem by a little peg. So if you look at the branches, you'll notice there's like a little peg that holds each of the needles individually to the branch. And then you can also distinguish the blue spruce based on their cones. Their cones, as you can see from the picture here, have a softer, more flexible texture to their scales, and they range from some around 1.5 to 4 inches long, or 4 to 10 centimeters. So you should be able to identify the blue spruce, as well as the next one, which is a Douglas fir. So the Douglas fir, one of the key features about the cones in particular that I always look for is that the female cone has this 3-pointed bract, this structure here outside some of the scales on the cone. The needles for the Douglas fir are 2 to 4 centimeters, individually attached, and the cones are 3 to 4 inches long. Okay, so slightly bigger than the blue spruce cones. These were a little bit smaller, but the Douglas fir are slightly larger, but not extensively so. Now, the eastern hemlock is distinguishable based on a very, very small cone. So I'll mention that first. You can see how tiny the little cones are here on the eastern hemlock. They are usually about an inch or less, really tiny size to the eastern hemlock cones. And then if you're looking at the tree itself, a characteristic I often look for is that it appears to have a drooping look to it. It's not a really strong pointy terminal shoot. It almost looks like it's drooping. And then if you examine the branches individually, there are the needles attached to the sides as opposed to all the way around in a whorl like we saw with the last two. See how the needles are all attached in sort of a spiral orientation on those, but in the eastern hemlock, the needles are coming out to the left and the right of the branch, so on the sides. Okay, and then also if you flip it over and you look at the underside of each of these needles, you'll see there are two little white lines present. So look for that if you're trying to identify the eastern hemlock from the branch itself. Okay, and the last one we have for this week for you guys to look at for gymnosperms is the white pine, and it is very easily distinguished based on its needles. You can see these needles are much longer than any of the others. They're also attached in groups called fascicles. Okay, so whereas all the rest had individually attached needles, I'll just flip back and show you, all attached directly to the branch, the pine has groups of needles attached in a bunch, okay, to the branch. And then they're much longer. Like I said, the needles are like 4 to 5 inches long. The cones are long and slender, somewhere around 5 inches, give or take a little. Okay. So those are the 4 examples we have for gymnosperms for you guys to look at this week. We're also then going to look at some angiosperms. So angiosperms are the largest group in the plant kingdom, and they are flowering land plants. We separate them out into monocots and dicots. Dicots are sometimes also called eudicots. And they have range of structural characteristics, including roots, stems, leaves, seeds, flowers, and fruits. Today, we are just going to look at the roots and the stems. In the next couple weeks, we'll look more closely at leaves and stomata, seeds, flowers, and fruits. Okay, so we'll look at those later, but today we're going to focus on roots and stems. All right, so monocots and dicots. Like I said, they're 2 species of angiosperms, and that name, monocot or dicot, like I said, sometimes eudicot, is taken from the number of cotyledons. So monocots have 1 cotyledon, and dicots have 2, di being 2, mono being 1. And that refers to, like I said, they're cotyledons. The cotyledons shown in the picture here are blue, and they are the seed leaf. So these are within the seed. It's a very starchy substance, a rich food source for the embryo. The plant embryo is going to be situated here, or in this picture here. And this is going to obviously need to develop for a period of time before it actually has put out its own leaves and can photosynthesize. So the seed, the embryo within that seed, needs to have a food source to sustain it for the beginning of its life when it starts to germinate. And that's what the cotyledon does. It helps to support the growing embryo before it can photosynthesize. Okay, so the cotyledon does that for it. And some seeds have 1, those are monocots. Some seeds have 2 cotyledons, and those are dicots. There are also a lot of other morphological differences between monocots and dicots, which I show here on this slide. So monocots have the top row here, dicots have the bottom row here. Like I said, their seeds have either 1 or 2 cotyledons, but they also have differences in the appearance of their flowers and their leaves, their root structure. And then if you were to cut open the root and look at the cross section, or cut open the stem and look at the cross section, you would also see a lot of differences between monocots and dicots. So just looking at the plant itself as it's growing, you'll be able to notice that the flower parts are arranged in groups of or multiples of 3. So for monocots, you might have 3 petals, or you might have 6 petals. And then these green structures coming off here, these are called sepals. You might have 3 of those or 6 of those. Versus for a dicot, you're going to have these flower parts in groups of 4 or 5. So you might have 4 petals, 5 petals, 10 petals, and so forth. So in multiples of 4 or 5 for a dicot versus multiples of 3 for a monocot. And then if you look at their leaves, monocots have a parallel vein orientation. So you see the veins running through the leaf here. They're all oriented parallel to each other. Versus in the leaf of a dicot, you've got this net-like orientation to the veins, okay, branching orientation. And then if you were to pull up the plant and look at the roots, the monocots have a branching fibrous root structure versus dicots have one taproot with branches coming off of it, one main taproot, and then branches extending from that. And then if we were to cut a cross section and look at the cross section of the root or the stem, we'll be able to see a lot of differences as well. And we're actually going to examine these two, the roots and the stems, for monocots and dicots under the microscope this week. So looking at the roots, you will notice that for a monocot, you've got your vascular structures arranged in a ring in the roots. So you'll see here a vascular cylinder where you've got the xylem and the phloem located there. Versus in a dicot, the xylem forms an X or a cross in that root. And then the phloem is going to be located in sort of the arms of the xylem. So phloem will be in here or in here, and the xylem will be forming this X. Okay. So again, remember xylem is responsible for water movement, phloem is responsible for food movement. Okay. And then if you look through the stems, same thing, cut it open, look at the surface. We'll do this under the microscope. You'll find out that in the stems of monocots, you've got vascular bundles. So your xylem and your phloem are scattered throughout the stem in different bundles, sort of scattered throughout. And then in the dicots, you're going to form rings, xylem and phloem, okay, within, like, so we're more towards the edge of that stem instead of just scattered around everywhere. Okay, so we'll look at those. Before we do, I do also want to mention a few different types of cells that are very common in plants. Parenchyma cells are the relatively typical unspecialized plant cells. They have thin cell walls, they're living cells. They make up a lot of the ground tissue. So basic structures of the growing plant. And then they can also store starch. They can also specialize. So in leaves, for example, if they have adapted to perform photosynthesize, then they might be called Sclerenchyma cells instead of Parenchyma cells. The next type is Sclerenchyma cells. These are actually dead at maturity. So they have a supportive role, a very rigid support element for the plants. Like I said, when they are mature, they have now solidified their cell walls as a support and kind of lost the internal organelles inside the cell. So that will form, these will serve as a support structure for the plant. And then Clancama cells are another support type cell, but they're flexible now as opposed to rigid. So they are present in young parts of the plant that are growing but still need a supportive element. Clancama cells are what we would recognize as the strings in celery and I think bananas, those like strings that you peel off. These are supportive, obviously thicker than the other cells surrounding them, but still living and still flexible. Okay. And then lastly, I want to mention xylem and phloem again. I know I had said xylem are the water conducting cells. There are actually two different types of cells, vessel elements and tracheids that make up the xylem. And for phloem, which is responsible for conducting sugar, there are sieve tube elements and companion cells. Okay. So for the vessel elements and the tracheids, these are also going to be hollow at maturity. They have a lot of open space to allow for water to be conducted through the cells. And phloem, the sieve tube elements are hollowed out shells of a cell, but they're supported by companion cells, which are living and use their organelles to support the sieve tube elements. Okay. All right, now let's look at roots and stems. First, we'll look at roots. Obviously, these are usually underground. They are able to absorb the water and minerals from the ground and take that up to the rest of the plant that needs water. You should be able to identify several different parts of our root. So first off, if we're looking at a longitudinal section of the root, at the very tip is the root cap. And this is when a root is growing through the ground, a lot of these cells are going to be sloughed off. They're going to be pushed through the ground and damaged and eventually shed and replaced by actively dividing cells from what we call the apical meristem, which is number 1 here. Okay. Lots of divisions going on in a meristem. Meristems are located in various places in the plant. The apical meristem is at the tip of the root here. Okay, so there are other, apex just means tip. In this case, we're talking about the tip of the root. Meristem is forming new tissue. Okay, and then as we continue up through the root, behind the apical meristem now, you've got the vascular cylinder in the center, surrounded by cortex. So the vascular cylinder is where we're going to find our xylem and phloem in the root of a monocot, which is what we're looking at here. And then cortex are going to be made up of parenchyma cells, general tissue for the plant. And then on the outside, we've got epidermis. And at various points, you're going to see, if you're looking like longitudinally, if you're looking from the top to the tip of this root, you're going to see different zones along the length of the root. So obviously at the tip, we've got a zone with actively dividing cells. So we call this the zone of cell division. So the meristem has a lot of cell division, adding new cell layers. Okay. And then so above that is the zone of elongation. A lot of the cells in this region are actually expanding and elongating, and that's actually what's pushing the root to a large degree through the soil. It's the elongation of these cells more than the production of new cells. The production of new cells are really just to allow for repair, and the elongation of the cells in this zone are actually what allow for the root to grow to a large degree. And then above that is the zone of differentiation or maturation, where you're starting to see some specialization, and you can observe that by the fact that these little root hairs are emerging to increase surface area for water and mineral absorption for the root. So you can see that's now typical of the zone of differentiation. Okay. And this is a root taken from an allium or an onion, which we'll look at actually under the microscope in lab this week too. We're going to look at it in long section, and then we're also going to look at it in cross section. So that brings me to this picture right here. So this is a monocot, and it's a root. This is a dicot root. The onion that we had looked at in the last slide is a monocot. We will also have an example of a dicot root for you, but this is what the monocot in cross section would look like with, as you probably remember from the last slide, the vascular cylinder on the inside, surrounded by cortex, surrounded by the epidermis. Okay, and I'm going to just zoom in a little bit so you can see this a little bit better. I want to point out the xylem and the phloem. So in our monocot root, the xylem is forming this ring of large openings, most towards the center, aside from this core. Okay, so this core is Parenchyma cells. It's called the pith. And then just outside of that, you've got these large openings that are our xylem cells. This is responsible for water conduction. Okay. And then just outside of that, you can see these little hazy spots here. This is another ring, but now this is phloem. Okay. So these guys are the cells associated with sugar conduction in the root. Okay. I'll also mention that there are single layers of what we call pericycle, and then just outside that, endodermis, you'll see here. A layer of cells that makes up the endodermis, and a layer just inside that that makes up the pericycle. And the pericycle is what actually produces those root hairs that we had looked at in our zone of cell differentiation. The root hairs actually emerge from the pericycle and travel out to the outside of the cell, or outside of the root, excuse me. And then I'll just zoom back out to show you just this is the cortex made up of parenchyma cells, the epidermis. The stele is often referred to as the section here, the vascular cylinder that contains the xylem and the phloem. Okay, and then we're going to move over to look at our dicot root now. So that's this picture here. A lot of the same structures are present. However, they take on a different orientation. As I had mentioned in one of our previous slides, the xylem is now forming this X structure in the root of the dicot, shown here in the top, and then enlarged here in the bottom. So xylem, and then the phloem is actually in the arms, within like the nooks of the arms of the xylem. So phloem here, xylem here. And then you again have your layer of pericycle and your layer of endodermis that make up your vascular cylinder. Then a lot of cortex made up of parenchyma cells and epidermis on the outside. Okay. All right, let's move on to stems. So stems, we know they provide structure for the leaves and for the flowers. They carry water, minerals, and sugar up and down. They can be characterized as either herbaceous or woody. So herbaceous stems are green, flexible, mostly found in annual plants, which will die off and reform year after year. And then woody stems are on those plants that continue year after year to expand. So for instance, you're familiar with a tree has rings within its stem. This is a woody stem. It has bark, firmer, needs to support the tree year after year. Herbaceous stems can be on monocots or dicots. Woody stems are usually found on dicots with a few exceptions. All right, so let's take a look at these in cross section. First, I'll direct your attention to the monocot. This is a monocot stem. And as you can see, you've got scattered throughout this stem these little bundles. These are your vascular bundles, and each of them have xylem and phloem. Okay, so let's zoom in just a little bit again to show you that. So you can see here, maybe in this one right here, you can see that there's some structures here. These are the xylem. The phloem is usually located just outside, more towards the outside of the stem than the xylem. So again, you probably have xylem here, phloem here. But they're scattered throughout the stem and surrounded by parenchyma cells. So all this ground tissue here, that's all going to be parenchyma cells. And then on the outside, you have your epidermal cells. Okay. And then we're going to look now at our dicot herbaceous stem, where you have, again, a lot of the same components, but they're oriented differently. So still have parenchyma cells forming a pith, the inside here, but now you've got these vascular bundles forming a ring, almost just inside our epidermal layer. So we're going to zoom in and look at those too. Okay, so here we've got our xylem and our phloem, and then a set of Sclerenchyma cells capping that. So for the Dicot stem, this is now a vascular bundle. And instead of having it scattered around like in the monocot, they're oriented in a ring around the edge of the stem. So again, this is xylem, phloem, and Sclerenchyma cells. Okay, and you might also see some patches of Parenchyma cells forming some supportive structures along the edges of the epidermis here and there to give some support to the growing stem. All right, I think that is it for that slide. Let's look at woody stems now. So woody stems, like I said, they need to support more than 1 year of growth for the plant. Obviously, typical of trees. If you cut down a tree and you look at the trunk, you see those tree rings, right? Those rings that have been added year after year. That's actually what you're seeing. What you're actually seeing there is the xylem that have been added a layer each year with every year's growth for the tree. So let's look at this diagram and then we'll look at the actual picture of a 2-year-old stem. So the diagram here in the top right picture is what a woody stem would look like after just 1 year of growth. Okay, so you've got your inside is the pith, and then your xylem most towards the inside, your phloem towards the outside, cortex, and then epidermis. So pretty simplistic after just 1 year of growth. But with the next year of growth now, we need to add a layer of xylem and a layer of phloem. And this happens via divisions that happen in this red line here, which is representing our vascular cambium. So cambium is going to be a dividing layer of cells, and it's going to produce an additional layer of vasculature. So the vasculature, again, is xylem and phloem. It's going to produce an additional layer of xylem just to the inside, and then an additional layer of phloem just to the outside of the cambium. So the oldest xylem is going to be most towards the center, but the oldest phloem is going to be most towards the outside because the new layer is added to each side of that cambium with each year. Okay, so the newest phloem will be, the secondary phloem will be just adjacent to the cambium on the outside, and then the secondary xylem will be just adjacent to the cambium but on the inside. I hope that makes sense. So that adds a year's worth of vasculature, an additional layer with each year of growth. And then you've also got, as the trunk expands, you're going to lose some of the tissue on the outsides. The epidermis is not going to be able to expand to the degree that's needed to cover the entire trunk with each year's growth. So there's also a cork cambium here, which adds layers to the epidermis that will then allow for younger epidermal growth. Then as time progresses, eventually the outer epidermis it dies and falls off. Okay, so that is the diagram of what the woody stem would look like. Let's move to our next slide so we can see what it actually looks like under the microscope. This is a tilia 2-year-old stem. It's a dicot. So right here, this dark line, this is your vascular cambium, which is adding the xylem to the inside and the phloem to the outside every year. So you'll notice here there's a thin line dividing the first year's xylem, the primary xylem, from the second year's xylem, the secondary xylem. Of course, it's going to be most adjacent to our vascular cambium. And then phloem is going to be to the outside of that vascular cambium, with the oldest phloem being most external and the youngest phloem being just adjacent then to the vascular cambium. Okay, and epidermis around the outside, pith and endodermis on the inside. And that is basically going to wrap up the talk for this week. So when you are in lab this week, we are going to be looking at the different clades of plants. We're going to have examples, hopefully, of mosses and liverworts and club mosses and all those guys up until I'm not sure if we're actually going to be able to look at a lot of angiosperms this week, but if not, we will look at those in subsequent weeks. And then we're going to examine the slides of roots and stems of angiosperms and be able to tell the difference in those between the monocots and the dicots. And then this week, there's a lot of drawing pictures in your lab manual, which I find that students are hesitant to actually do because it's a little bit time intensive, or they don't think they're good artists. But I do think it's very helpful for you to learn the life cycle based on what you're seeing and be able to identify which parts of the plant are, say, the gametophyte stage and which are the sporophyte generation. So I certainly encourage you to spend time drawing those, even though you might not think you're a great artist. It's more for you to process it than to get great pictures. All right, so that's it for this week. I will see you in lab on Tuesday. I hope you guys are all doing well. And if you have any questions, don't hesitate to shoot me an email. I'll see you soon. Take care.